So good morning. Thanks for all being here, and thanks for having me. I'm an advisor to Active Insights, and I'm a physician. I've actually been a physician for a very long time, over 45 years. In the last 6 years, I've spent exclusively in the digital biomarkers, real-world data, wearable space, and it's really given me a new perspective. In fact, I've had two major epiphanies. The first one is that every disease is a systemic disease. A patient with a disease responds to it. Their whole body responds to it. Their immune system responds to it. Their autonomic nervous system responds to it. Their activity is affected. Their sleep is affected. If you look for the sign of the disease, you will find it. So a million years ago, when I was an intern, I had a clinic patient who was a very heavy smoker. And I used to tell her, please quit every time I saw her. And she's like, I don't know. Why should I quit? I feel fine. And I said, oh, come on, you can't tell me you're not short of breath when you go up a flight of stairs. And she looked at me and she said, I never go upstairs. And there was born the first digital biomarker, stairs climbed as an index of chronic pulmonary disease. The second thing is that, I hate to say it because my whole career has been involved in this, we've been doing it wrong the whole time. Seeing people every month, every 6 months, God forbid, once a year for your annual checkup, this is not the way to either promote health or to follow disease and its treatment. I'm not the first person to have this thought. In the inaugural issue of Digital Biomarkers, the founding editor cited the social physics principle that we should be making observations per person per minute, not per month or per year. And that this is based strongly on the scientific Nyquist principle that says that the variation in the system, maximal, should be exceeded by twice that by the sampling rate. Well, when you think about how complex a human being is and their response to disease, it's pretty obvious that the answer is continuous monitoring. That was picked up on by the Harvard Business Review, who actually had the bold statement that the future of drug trials is better data and continuous monitoring. And they even coined the term Continuomics. This report from the University of Oregon kind of proves what we're talking about. How is a better way to follow disease, detect disease, follow therapy in patients? Simple. They did a large study and they said, what if I wanted to see a 30% effect size in cognitive impairment in elderly patients and measure the way we usually do with an annual physical and a memory test and a mini mental status? You would need 2,000 patients in 4 years. They said, now what if we did it with something that was continuous, that correlated with cognitive decline, and let's use walking speed, for example. If that were the case, you needed half as much time and only 200 patients, 1/10 the size of the cohort. Now let's take it a step further and let's make it, in the context of use, let's make it on point for cognitive functioning and do something like count mouse clicks or web pages visited. Now you need 26 patients. This is where we can go with digital biomarkers. So what should we look at? Vital signs, behavior, lifestyle, activities, sleep, etc. Yes, I'm going to emphasize how those things become important viewed through the lens of a particular disease, but I also want to talk about disease-specific clinical signs and functional outcomes. This is a list of diseases and TAs, and what I'm trying to do is say, look, vital signs sounds generic, sleep measures sounds generic, and they are. But when they are viewed through the lens of a particular disease, they take on disease-specific characteristics. So, is there a sleep disorder in Parkinson's disease? Yes. Is there a sleep disorder in PTSD? Yes. Are they the same? No. The active insights experience echoes this. This is in the commercial world. Incoming interest in developing digital biomarkers, research projects, collaborations, publications, et cetera. And what you see here is that out there in the real world of commercial interest, almost every TA has been explored for digital endpoints. Let me give you a reason why. Let's look at this two different ways. Let's ask the question, what if we had one measure and 3 different diseases? And then let's look at what if we had one disease and 3 different measures. This publication indicated that activity levels in patients just discharged with heart failure correlated with rehospitalization and mortality, the hard endpoints. This is a study indicating that activity, this is step count, stairs climbed, et cetera, correlates directly with the Karnofsky Performance Scale, which is usually done subjectively in oncology patients. This is a population of patients with multiple sclerosis. Now in MS, when you cross EDSS, that's the scale that they use, subjective mind you, when you cross 4.5, you need assistance. But when you actually look at the population who has an EDSS less than 4.5 and one that has more, the difference in the population is 5,000 steps per day. On the subjective scale, it's half a point in the mind's eye of some other person. Whereas we could measure this with digital precision long before the final clinic visit when somebody said, hey, do you really need a cane? And they said yes, and they said, oh, I guess you're a 5. Now let's look at it differently. Let's say we're just looking at advanced cancer. Instead of the Karnofsky Performance Scale, here we have the ECOG score. I'm going to come back to that in a minute because you see how wide these error bars are? That's not because the activity measures are so screwy. It's because the subjective decision to make somebody a 0 or a 1 was just that. And when you actually examine them functionally, you find out that it's very disparate. And I would submit there would be a much better way, in fact, to do functional assessment of patients rather than the subjective. Now, this middle graph is an obvious Kaplan-Meier curve, and it's in advanced cancer. So when I see that, I say, oh, this must be surgery and radiation and chemo. No, it's not. It's autonomic tone. It's heart rate variability. The patients who are compensating, who are acclimating, who are winning the battle with their disease have more normal autonomic tone. Those who are more at risk for dying do not. Over here, we have another set of Kaplan-Meier curves, again, highly statistically significant. Again, not surgery versus radiation, et cetera, et cetera. This is sleep efficiency. Of the time you spend laying down, how much of it are you actually asleep? When I presented these data to a large CRO, somebody said, oh, come on, there's a lot of reasons patients with cancer don't sleep. I said, yeah. Let me give you another example. And this is where I had an epiphany. This was December 2015, New England Journal of Medicine, isosorbide mononitrate in heart failure with preserved ejection fraction. You've seen this article 1,000 times, and you expect that what they're going to do is say, well, the stroke volume, the ejection fraction, the cardiac output, they all know. The primary endpoint was the daily activity level. Now imagine that. They gave a drug to a sick population that was well defined, and they asked, how's the patient doing? How's the patient functioning? Now, believe it or not, this is very good medicine because you can't have activity unless you have good blood flow, you have good oxygen delivery, you have good cardiac output. So if indeed the drug was working and improving the delivery of oxygenated blood to the whole body, then activity would go up. And if it didn't, it didn't. So it's not that they weren't measuring cardiac performance or response to this drug. It's that they actually took the patient's perspective and looked at a functional outcome. Here's another example of something very simple and yet very powerful. These data come from the University of Rochester. 4 populations, controls age matched, patients with Parkinson's disease, patients with symptomatic, that is motor symptom Huntington disease, and patients with prodromal Huntington disease. We know it's a genetic disease. You know it since birth, that you're gonna get it at some point, but until and unless you have so-called motor symptoms, you're considered prodromal. In the 3 groups, control, Parkinson, and prodromal, people spend about a third of their day laying down. Patients with symptomatic Huntington spend over 50% of their day laying down. Now, why is that? Well, it turns out that, first of all, chorea is exhausting. Chorea is a fall risk, and chorea gets in the way of you doing anything that you want to do. And so how does the patient mitigate this? They lay down. Now, if I was a drug company and I had a drug that I actually thought would help with chorea, all I would have to do is look at their posture over time and see if, in fact, it didn't return towards what everybody else does. And similarly, if I had a drug and I was going to intervene, I was going to do something drastic like a gene therapy, wouldn't it be nice to know the minute that they started to look more like symptomatic and not wait until they were at this advanced stage before intervening? So this is some of my favorite data, and I just heard the panel up here talking about how sleep can be a challenge, but this is a little bit of fun. I already showed you sleep efficiency in oncology. This shows that the only difference in life that was statistically significant between dementia with Lewy bodies, dementia with Parkinson's, was sleep fragmentation. The only other 2 things significant were at autopsy. That's how powerful it is. But this is the one I want to focus on. This is 100 patients with inflammatory bowel disease. Every one of them got an endoscopy, every one of them got a biopsy, every one of them had a histology score. All of the patients who had active histologic disease went to the sleep lab, and 100% of them had sleep disturbance. Of the patients who had histologically inactive disease, that is to say, no sign of activity, no sign of inflammation, they had a normal biopsy, went to the sleep lab, and half of them had a sleep abnormality. And now for the punchline. The half that had a sleep abnormality all had relapses in 6 months. Your body knows. I don't care if you can't even see it through the microscope, but something is going on at the microbiological level, at the genomic level, at the proteomic level, stuff that you can't know, but it becomes manifest clinically in the patient that's diseased, and if we look for the signs of it, we will find it. I told you we'd talk more about this, the activity in heart failure. In this first study, activity correlated with the hard endpoints of hospitalization and heart failure, but it also, hospitalization and mortality, but it also correlated p less than 0.05 to all of these other things, including hospitalization, but things as arcane as pro-BNP. Now, BNP is secreted by your body when? When the transmural wall tension on your left atrium goes up and your body thinks you are in worsening heart failure, it secretes pro-BNP. Pretty cool biomarker. But what you're not going to do is get a pro-BNP level in your living room or every day, whereas the step count can be done every day and in your living room and has the same predictive power. So powerful, in fact, that in this study, this is a sick population of patients with heart failure. These patients all had either an implanted defibrillator or CRT, cardiac resynchronization therapy. They were monitored for the first 30 days post-hospital discharge. Patients with reduced early physical activity had increased hospitalization for heart failure, increased mortality. Look at the p-values. Look at the p-values. So much so that each increase in physical activity of 10 minutes resulted in a 4% reduction in both death and hospitalization. So how much more powerful does a digital biomarker need to be than that? So I'm going to kind of leverage some of what you've heard before, but put a kind of clinical spin on it. 20 years ago, this Gait signature was published in an engineering journal. You know, the heel strike, the toe off, the leg swing. This is the signature of normal gait. In 2015, this was reproduced with a wearable sensor worn on the anterior tibialis muscle of the leg. So this is something you needed a kinesiology lab to do. This is something you can do with a stick-on accelerometer on your leg. But so what? That's the important question. Just for the question for the day, the important question to ask is, so what? I can measure this. So what? Well, here's the answer. If you look here, you see in the background that signature of so-called normal gait. And you can compare it even visually to this disarray, which was the gait of a symptomatic Huntington patient. When the patient was given tetrabenazine, this is what happened. Not only can we see the drug effect, not only can we see the way in which the Gait signature has returned toward normal, and that can probably be mathematically quantified, but the punchline again is we can do this in your living room. We can know if your medicine's working. We can know if you're taking your medicine. We can know if it's actually helping you. This is Parkinson's disease. You've heard of bradykinesia, so-called slow movement. Well, when you move slow, what ends up happening is that the length of time each step takes is long. So here's a patient with a long step time, and at the dotted line receives L-DOPA. And now step time is markedly reduced. So what's so good about this? Well, there's three things that are so good about this. Number one is we can digitally precisely characterize bradykinesia and actually see it dissipate on treatment. Number two, and it's very important in pharmaceutical development, you can tell when the patient's off medication and when the patient's on medication. And the best thing of all, these data are from somebody's living room. They were acquired from the home. One more example just for fun. This is, again, Parkinson's disease, off medication. And what you see here is the tremor being characterized by its frequency power spectrum. There's a bright light here at 5 hertz, and there are even harmonics at 10 and 15. Cool. I would challenge any clinician in the room, myself included, to be able to see this with the naked eye. But it's going on. It can be measured. After L-DOPA, look what happens. The power spectrum is reduced at the principal frequency of 5 hertz, and the harmonic overtones went away. This is something no clinician can tell you, no laboratory can tell you, and yet this can be done in the home with wearable technology, digital precision, and clinical significance. So the evolving solution. Real-world data are increasingly being used in clinical development trials, as are functional outcomes, and the precedent has been set for a qualified functional disease-specific endpoint. I was happy to see Jen point this out because this was the icebreaker. Somebody finally approved a digital biomarker for a disease-specific endpoint. Excellent, right? This is what we mean by context of use. You can strap an accelerometer on an ankle and measure a whole bunch of things, but in this particular case, it turns out that this is the relevant metric for people with Duchenne, and that's where we need to go. We have the technical capability. We now need to have the clinical focus and the context of use to pursue digital biomarkers. That's what this whole process is about. We already know how accelerometers work. We have analytically validated them. In some cases, too many metrics, too many for motion, for example, or activity. But what we need now is What does the step count mean? Well, in the heart failure patient, I think I showed you what it means. In the cancer patient, I think I showed you what it means. In the MS patient, I think I showed you what it means. So this is where we need to make our mark with digital biomarkers, is in the context of use piece, which will need to be clinically validated. Active Insights has already characterized the technology, has already characterized the algorithms and the capabilities for biometrics. Now it is time for that third step, which is to find a context of use, a clinically meaningful difference, and a legit biomarker. We're getting there. As you heard Jen say earlier today, more than 100 pharma companies, in fact, have included more than 360 unique digital endpoints. That's actually pretty cool. And more than half are in drugs and another third in devices. And yes, it is remarkable. A couple of years ago, most of these would have been exploratory. Now, believe it or not, over 100 are primary and 200 are secondary endpoints in these trials. This is real progress. This, on the other hand, in my opinion, is not. We have a qualified COA program at the FDA, and it's active. There are over 72, I think, at last count, applications, but only 8 are for digital health technology, and more than 75% are for PROs. Now, this is going to be very unpopular, but I'm going to say it anyway. Digitizing somebody's subjective opinions is not a digital biomarker. I'm sorry. Well, this just in, to give you an idea of how pervasive this thought about activity monitoring, this is from just last month. Cancer and cardiovascular disease, incidence, mortality, and all-cause mortality, and incident dementia in a population of over 78,000 in the UK. So the world is evolving to accepting the idea that, hey, measuring steps, measuring activity, it really does make a difference. And as I promised you at the outset, if you look for it through the lens of a particular disease, you will find it. Now this says that the number one thing that predicts your longevity is gait speed. Not hypertension, obesity, diabetes, smoking, ethnicity, nothing. Gait speed. So keep moving. You slow down, you stop. I have 2 more I'm going to show you just for fun. We know that if you use a selection biomarker for a clinical trial, you get better results. Phase to phase, and from first phase to third phase, triples if you use biomarkers. Well, whatever happened to this ECOG score? Look at the error bars. Somebody said, I think this is a 0. Somebody said, I think this is a 1. But when you look at them functionally, they're all over the place. I think the selection biomarker should be a functional digital biomarker to decide who should be in a trial. This is where I'm going to leave you. This is the natural course of Huntington's disease. When you have a so-called motor diagnosis, they start measuring you on the UHDRS. Meanwhile, look at how much brain matter you've lost. But here's the punchline. By the time you register on the UHDRS, you have lost 30% of your brain volume. Look at the size of these ventricles. Look how much white matter is lost. Look how tiny the basal ganglia are. 30% of your brain is gone before you rate on the subjective scale. The point I want to leave you with is it's time to write the natural digital history of disease. We can do a lot better. Thank you.
That was a great note to end on. All right, thank you so much. So my favorite parts of that were really the move towards the functional assessment of patients and outcomes, and the ability for digital measures to look for clinical manifestations of disease before patients meet the predefined threshold of disease diagnosis. There's so much potential within digital. And he harped on the fact that digital measures must be objective and not subjective.